Hello, everyone. I am Jeff Galvin. I'm the CEO of American Gene Technologies, and we will be getting started more momentarily. There we go. Just wanted to make sure that I can see what was going on in my screen, which I can. And we have a very good audience today. Thank you all for coming. This is a press conference that we're putting together to help explain what is going on with our HIV Cure Project. I'm sure all of you are aware that we have been working on this for quite a long time. It's actually been a goal or a dream of the company for almost a decade to find a way to cure HIV using gene and cell therapy. And since that time, there's been a lot of hard work and also a lot of serendipity. We had started off the company just to leverage the power of gene and cell therapy to cure a myriad of different diseases, because I saw this coming from a different industry, coming from the software industry, as being a new kind of component architecture in drug development, where you could make a tool set that you could use with a whole bunch of different components that you could mix and match to cure a myriad of diseases. And then we were just going to look for low-hanging fruit that was curable with this progressing set of tools that we were going to develop at American Gene Technologies. And as luck would have it, there was some interest at NIH to test a theory that we had about HIV. And they gave us a small grant, which we added to the money that we were investing in that project. And we were able to get to an interesting result with the help of somebody that is also on this conference, David Pausa. So he'll be joining us in a moment. David Pausa at the time was at the Institute of Human Virology. So this was the first part of the serendipity, is that only about 45 minutes north of where American Gene Technologies is located in Rockville, Maryland, is the Institute of Human Virology, which is founded by Bob Gallo, Bob Redfield, William Blattner, in order to just continue the study, the research, and the treatment of HIV. And it's one of the top institutes in the world in research and in the clinical application of therapies that have really ended the issue with people dying from HIV. It is, through their work and the work of many other great researchers around the world, they had converted HIV from a from a death sentence into instead just a lifetime of taking a suppressive therapy, but that patients would be able to achieve a relatively normal lifespan. And that was remarkable for any of us that remember how scary HIV and AIDS were during the 80s. That was a major step forward. But in any case, they're located not far from us. And David Pausa was a consultant on that project that helped us to get that initial grant from NIH. And the results were pretty good. We got 3 to 4 log reduction in viremia in an HIV challenge test using a gene therapy to protect the T cells. And I think that further intrigued not just David Pausa, but Bob Redfield, who I'm sure you're all familiar with at this point, who was at that time the director of the Institute. And the two of them were gracious enough and excited enough about what we had envisioned that they jumped in, and it was largely through their encouragement and also an incredible scientific input by David Pausa that we were able to move from just that proof of principle into a practical approach to changing a HIV-infected individual's immune system so that they could be immune to their own disease and to control it naturally with a recovered HIV-specific T-cell population that we could protect and enhance in the body. And I can't tell you how much, just amazing science went into it and how many details that David Pausa and the team that he put together here at AGT, went through all of the efforts and research and all the problems that they solved to come up with something that I think has a tremendous probability of being a cure for HIV. Now, that's not a guarantee, but I can tell you from being in gene and cell therapy now for 12 years that I have watched the industry, I've watched these projects, I see the trajectory of these things, and I see the milestones. And I think that what we have right now is the ability to take 400 milliliter leukopak from a well-controlled HIV infected individual, HIV positive individual that is well controlled on antiretroviral therapy. So that means they have to have been on ART for somewhere between 1 and 3 years. And within that 400 milliliter leukopak, we can find enough HIV specific T cells that we can isolate them, modify them so that they're completely immune to HIV, amplify them up to about 1 billion of those cells. And the specific cells that we are looking for, isolating, and enhancing are HIV specific CD4 positive helper T cells that are responsible for initiating the entire immune cascade against HIV. And by returning those billion cells that are immune to HIV into the patient, we think that this could give them what's called a functional cure. Functional, I like to say for the layman, means as good as, but it's a technical term, but what it means is there may be some virus still in the body hidden somewhere. You know that HIV is so insidious because it can hide all over the place in the body. But the key is that the patient wouldn't need antiretroviral therapy anymore to control that viremia. Their natural T cells that are specific to HIV and their normal immune response to HIV would suppress the virus at low enough levels without antiretroviral therapy that they could not give this, not infect another person with that HIV. They could never progress to AIDS. They would be basically at the undetectable levels that you hear on TV. Undetectable means uninfectable, U equals U. So we could get them to that level of control using their natural immune system with this enhancement that we're able to give in this cell therapy. And I'm sure we'll get more into the technical details later, but I think this is something that will make HIV people feel normal, both physically, and also I think another important impact of this is psychologically. In other words, they would know that they were of no danger to anybody around them that they love. None of the people around them would have any reason to be concerned about anything that is in that person's body. It could not be transmitted. And they would know they would never get AIDS. I think the daily benefit would be that they wouldn't be taking that daily antiretroviral therapy, which would mean that they wouldn't be suffering the side effects of that antiretroviral therapy. And what are the side effects? Well, you can just see the Biktarvy ads or any of the antiretroviral therapy ads on TV. Common side effects include nausea, diarrhea, headaches, fatigue. And then the long-term side effects can be liver, kidney, heart disease, extra cancers, early aging, bone density issues, osteoporosis, brittle bones. In fact, it turns out that insurance companies have a very expensive burden maintaining HIV patients. Even though these HIV patients will have a normal lifespan, they'll spend a lot more time in the doctor's offices for those side effects of the low-grade viremia and also the toxic side effects of long-term exposure to a chemotherapeutic drug, the antiretroviral therapies. So getting rid of that daily antiretroviral therapy is a major improvement in quality of life and also reduction in cost of maintaining that patient. So how much can the insurance system save? Probably $80,000 to $100,000 per patient per year for the patients that are being treated for HIV. Now, here is the thing that really shocked me when I found it out. that out of all of the people in the United States who know they have HIV, approximately 1 in 4 is taking antiretroviral therapy. And there's a variety of different reasons why they might not take antiretroviral therapy, but I think that a big one, of course, is the just severe damage to quality of life with that daily antiretroviral pill. And I think some people just put it off as long as they can. They feel normal. They're not getting pneumonias or other symptoms of AIDS, and they figure, okay, they'll get on antiretrovirals when they really need it. But the problem, of course, is that means that 3 out of 4 people in the United States who have HIV are still contagious. If everybody would be well controlled on antiretroviral therapy, we could reduce the spread. And the spread is quite a serious problem. The spread is 30 to 50,000 cases a year in the United States of new cases. Keep in mind that polio at its peak was only 60,000 cases a year. So this is an epidemic, and it's worth thinking about it from that perspective. But it looks like we can cost effectively take these patients, not only prevent them from being contagious, not only prevent them from getting AIDS, not only reduce the cost of maintaining them, but and also get rid of the daily and long-term side effects of antiretroviral therapy that are causing this major reduction in their quality of life. But we believe that this may be a one and done treatment that doesn't just cure them temporarily, it cures them for life, that they cannot be reinfected with HIV. We think there will be enough CD4 help maintained in the body that even if this treatment were to eliminate all of the HIV in a patient's body, that the CD4 help would remain around in sufficient quantity that it could easily reconstitute itself if the patient were to be exposed again. And this would just be amazing. I mean, amazing. Think of how you personally would feel if you had this enemy within. It's like a cancer that you can never get rid of. And it's contagious. You may be very careful not to infect anybody else, but there's a stigma surrounding that. It's a stigma that patients feel, and it's a stigma that society feels about HIV. You know, you see yourself, or you could see yourself as a little bit of an outsider. It's not the kind of thing that you just, feel open about. It's not like hair color or even skin color, which, can, in some cases, can evoke a stigma or a prejudice response. No, it is, something hidden away inside of you that is not the kind of thing that you just share openly. And so it's just forces these people to have a secret that they carry around. That's not going to be a great feeling, I imagine. I think that the ability to know that you're basically normal, that nothing is there, that would be a threat to anybody else, is going to be a major psychological benefit. But then also knowing that the enemy within is vanquished, right? Think about this: the viremia is gone, the infections, the potential of infecting somebody else is gone, the chances of progressing to AIDS is gone, the meds are gone. You're really, really normal. You're actually super normal, because you can go back to a community where perhaps it's a vulnerable community where HIV is still a problem, but you don't have to worry about picking it up again. I mean, this is just tremendous. And I think this is what the community's been dreaming about. It's what AGT has been dreaming about. And it's something that I think that we just achieved an incredible milestone that says that we're going to get a chance to see if we've reached that threshold of functional cure. We're going to be able to start the clinical trials now that the FDA has been satisfied with the safety data and with the clinical protocols that we have presented to them, and they have released us to start the clinical trial. So this is a very exciting day for AGT. We are, cautiously optimistic But I got to say that my personal confidence over this is quite high, and it really boils down to the dream team of HIV people that have gelled around this project. And I would suggest that all of you take a look at our website and really dig into the caliber of the people that have worked on this project and the backgrounds, and also, I think, a very important aspect, their passion for finding a cure. These are folks that have dedicated the last several decades of their life to get to this day. And I think that there isn't another more caring group of people on the planet and more empathetic group of people on the planet than our team around this AGT project. And this is, of course, from the scientists, but all the way down to the janitor in this company. This is the goal of the company was to use gene and cell therapy to reduce suffering and reduce death from serious human diseases. And now we're about to address something that, or we have a possibility of making a major leap forward on, a disease that 40 million people, nearly 40 million people on Earth have, and 1.1 million of our fellow American citizens have that disease. So this is just a turning point for the company. We think that it's going to be a turning point in HIV treatment and HIV cure. And that's why we're here today. We just want to give you a chance to ask us any questions that you have about the future of this project, the likelihood of this thing working, the schedule of the upcoming clinical trials, information on the data that we've gotten so far, and anything else that might come to mind. Now, this is a press conference, and I do understand that there are a number of people that are in here that are not press, and we are interested in getting to everyone's questions, but we're going to give priority to press questions. So if you don't think we would recognize that you are press, and if you are press and you want to get some priority for your question, please mention your publication in the question and just put it into the chat. And anybody can put something into the chat. We're going to be watching that for questions, and you can go ahead and start on that right now because we're going to move over to questions relatively quickly. And the next thing to do is just to introduce you to David Pausa. David, are you there on the line? Can you flip on your camera and join us?
I am here.
All right. So David Pausa is the CSO, Chief Science Officer of American Gene Technologies. And I told you about the serendipity of meeting David quite a long time ago. And how many years has it been now? About 5 years since you pretty much got on this full time.
Yes.
But it was really remarkable. I think it was a credit to two things. One was the trajectory of the project at the time, that it really looked like something that could work. And one of the things that David said to me and his colleague, Bob Redfield, said to me was that they thought David could do more to cure HIV at AGT than even at the Institute of Human Virology, which is just, what an incredible, confidence for him to exhibit in the company. His moving down to the company really formalized all of the science, and getting him on this full time has been the thing that has brought all the pieces together. It's changed it from a theory into something where we were able to think from solution back to our technology and create a process that is cost efficient enough and scalable and distributable so that commercialization is really built into the project. So, one of the exciting things going forward is if we get first human efficacy in this upcoming study, this phase 1 safety and efficacy study, if we get an efficacy signal, and especially if we can get, proof of concept, well, this thing it's run in a benchtop unit that costs about $250,000, that doesn't need a clean room, that could be purchased by any hospital or any clinic in the world. And if they can do a leukapheresis to get blood from HIV positive individuals that want to be cured, and they have a way to do the reinfusion protocol. So almost all hospitals and clinics could handle this. They can start treating patients. And what they have is an automated cell protocol inside that benchtop unit. It does need a special operator, but it's basically, you know, push a button and it works on these things for 11 days and the cell product comes out the other end. So they could be in a very reliable and practical way treating patients almost anywhere. It doesn't require a central processing facility. And I think that's very important to getting this solution out to the world. And the other thing that is really astounding about the preclinical data is that out of about the last 12 runs that we've done of this automated cell protocol, all of them but one have worked. Now, this is, if you're not familiar with CAR T, this is one of the things that held up CAR T, is that it is a larger procedure and it isn't, it doesn't meet the release criteria as often as 9 out of 10 times. And, but these things at this point are making what we think will be a working dose, a effective dose of these HIV-specific CD4 positive T cells 9 out of 10 times from the patient's blood. And the patient just needs to be well controlled on antiretroviral therapy, no symptoms for the study, it'll be no symptoms of AIDS, and a CD4 count of 500, which is not overly ambitious. So, you know, there's probably a couple 100,000 people that are in that range within the United States right now. So, and, you know, we can go ahead and do that at a price where the insurance companies will be saving money on the cost of maintaining those patients. And so I think that this will get out there. There'll be demand for it. I think this is really, you know, something that the entire market is dreaming of. And I think we have a practical solution. And now with the FDA clearance, we're going to be allowed to step forward to see if we can prove this in the only valid model for HIV, which is humans, because no other animal gets HIV. The H is for human. No other animal gets the same manifestation of HIV, no matter how good the model is. So this is really the gold standard. This is the only way to really prove it. We have tremendous confidence because we've made these blood products and we can see how well that blood can clear viremia and latently infected cells. And we can just do the mathematical comparison to the body, and it looks like it is more potent than it needs to be in order to get into the body and maintain the suppression of the viremia for life. So, okay, with that, David, can I turn it over to you? Is there anything that you would like to add just in terms of overview? And in the meantime, I'll take a look at the questions and then we'll pick from those questions.
No, I think you did a great job, Jeff, just introducing the whole situation. I would say that as this kind of a clinical trial rolls out, it's operating, of course, with the full advice and consent of the FDA. And the FDA has a very serious level of concern over patient safety, and especially over the safety of HIV positive individuals who are stably suppressed with their virus on therapy. So we've had to meet an extraordinarily high bar for entry of this study and for approval of this study. And that high bar covers everything. It covers everything from advising trial participants of potential risks that would be involved in the trial, all the way up to the methods for recruiting individuals and how long between individuals we have to wait. If we enroll one person and we infuse them with cells, how long do we have to wait to make sure that product was safe in that first volunteer before we move on to the second one? So this is a trial that's extraordinarily leading into participant safety. Our recruitment procedures are incredibly conservative and will focus mainly on the clinics that are hosting the trial sites, which so far are going to be a site in Northwest Washington, D.C. called the Washington Health Institute and the Institute of Human Virology at the University of Maryland, Baltimore. And if necessary, we are prepared to add at least one other site here in the Washington, Baltimore metro area.
Okay, thanks, David. All right, well, let's go ahead and move into questions. We have a little bit of a challenge here from Richard Jeffries, Treatment Action Group. Hi, Richard. Your press release states AGT believes there is a high likelihood that participants in the upcoming trial will be cured when it's not an efficacy trial, but a phase 1 first-in-human trial. Do you feel it's fair or ethical to state that confidence publicly? So, look, we're, basically just so people know, that's not exactly what we said in the press release, but yes, we are quite confident about this treatment. Now, it's a phase 1, but remember, it's being done all in HIV positive patients that are that have the attributes of the patients that this treatment is intended for. So we do expect to not just get safety data from this, but we expect to get efficacy signal. And if you look into the specification of the clinical trial, it'll be up on clinicaltrials.gov fairly soon, because we expect to start recruiting in September for this. What you'll see is that we have markers of efficacy as a secondary endpoint. We do have a lot of confidence in this treatment. Let's compare this to some of the other treatments in the past that have worked. One is Sangamo. I mean, let's give them some credit. There were some things that were showstoppers in their project. They were using zinc finger nuclease to clip the CCR5 gene to reduce the expression of CCR5 on the surface of the cells to make them more difficult or impossible for HIV to infect. Okay, so that's a good theory. That comes from the Berlin patient and comes from the London patient, that people that are CCR5 deficient can control their HIV naturally. Their body can fight it off. This is the whole theory behind our immunoregulatory approach. If your HIV T cells cannot be infected, if they can't be infected and depleted by HIV, well, then they do their job, right? They will actually clear HIV. And this is the way, your body works to clear a cold or a flu or whatever. The evolved advantage that HIV has had over the body is that it can infect the sentinel T cell that's supposed to detect the pathogen and start the immune cascade. If you can prevent that initial infection, then the HIV T cells can do their job. And what we're doing is we're taking a rare genetic mutation that occurs in reasonable quantities in Northern Europe, that CCR5 deficiency on the cell, and we're giving it to HIV infected individuals who don't have that deficiency. So the Sangamo theory was the same. And they use zinc finger nuclease, and one of the issues there is that when you go to clip genes, you have to get both alleles, otherwise you get plenty of CCR5 for HIV infection on the T cell still. So you can only drive the reaction up so high. It looks like maybe they were getting 1 in 100 cells where they were clipping both alleles. Then they actually got a patient that was in remission. It turned out that patient was what's called a delta 32 mutant. They were already missing 1 allele of CCR5. So they were getting, in that case, they would get about 1 in 10 cells with their zinc finger nuclease clipping, and that turned out to be sufficient to give that guy a durable response. This is very important. That was an important data point in establishing this overall theory and in also giving us confidence about our approach to this. So the first problem was sort of the reliability. The second problem was the amount of viral vectors, $500,000 per patient. And this would only work on people that were delta 32 mutants, as far as they could tell from the first phase 1. And so they did another phase 1. And this time they recruited all delta 32 mutants. Unfortunately, they still only got 1 in 10 in durable remission. So what this showed is that you'd have to spend about $5 million worth of vector and treat 10 patients in order to get 1 cure. Obviously, this was not a commercial project, despite the fact that it was a very important scientific finding. Okay, so how have we corrected that problem? One is our process only uses $35,000 worth of vector. So it's a fraction of the cost of what they were doing. It uses an siRNA knockout. We install a gene that creates a short interrupting RNA, actually a short hairpin RNA, out of a microRNA scaffolding that when the microRNA breaks down, it turns into antisense strands that will block the message from the CCR5 gene, and it doesn't matter which allele it comes from. So we're getting 100% in every cell that we can transfect within our cell process. They're getting all of their CCR5 stripped. And guess what? This is all proprietary technology that David and his team have developed, but we're getting 90% transduction rate in the sample of T cells. And we're also depleting out T cells that aren't of interest. So we're really focused on the HIV-specific CD4 positive helper T cells, which are the critical ones to restore immunity to this disease. So we have sort of boosted the reliability. We also added siRNAs against conserved regions of additional strains of HIV that don't need R5, the CCR5 to get in. And what that means is that even X4 tropic strains will be prevented from infecting well, they could infect still, but they wouldn't be able to integrate with the genome. And if they integrated with the genome, they couldn't express. So essentially, we're giving them protection, not just against CCR5 strains, but also non-CCR5 strains. So that was another sort of boost in the reliability or the broadness of it. But here's the kicker. If you look at the number of these critical cells that are in the bodies of people that came from the Sangamo study and are in durable remission without their antiretroviral therapy, if you measure the number of HIV-specific CD4 positive T cells and you compare it to the worst case of our cell product, it is 2,000 to 1. So not only are we getting 90% of the cells getting 100% of the CCR5 stripped off, but we're returning 1 billion cells, which is about 2,000 times better than the best case in Sangamo. So I think this will get us across the threshold. So we're big believers in it. I'm sorry, we're optimistic, but we've never promised that this is a cure, and I'll look you right in the eye right now, and I'll tell you I can't guarantee it, but I also will tell you that I'm highly, highly confident of this. And it was the HIV challenge data that finally convinced me. When you go ahead and take our cell product and you dump latently infected cells in there and you raise the viremia, it has a remarkable ability to clear itself of the virus and of the latently infected cells. And I think that you're going to find, if you look at the data that's published in Molecular Therapy, the article that came from NIAID, where they repeated our experiment on their own patients, I think what you're going to find is that when you look at the clearance capability of those tests, it looks like it's more potent than it needs to be to clear it in the body upon reinfusion. So that's why we're so optimistic. And I, as the entrepreneur and CEO of this company, I say confident. So I think it's not unfair for us to say that we think we will get some efficacy signal in the phase 1. We are going to study markers in the blood, and we hope to convince scientists around the world that we've got it, even if, because we don't call for a treatment interruption in this protocol, but even if we don't get any of these patients to throw away their meds and that we can verify that their viremia doesn't rebound, we think we will be able to find surrogate markers that show that these T cells are in there, they're healthy, they're active, and they're reducing viremia, and that it's highly likely that even if the antiretroviral therapy was withdrawn, these people would remain suppressed. So that's the CEO, 30,000 foot, unqualified from a molecular biologist, immunologist, virologist, HIV specialist point of view. I'm going to turn it over to David. If you don't mind, David, for a minute, either back me up on what I said, nuance what I said, correct what I said. because I want to be scientifically accurate about what we're giving to our friends over at TAG, who, by the way, we follow you all the time, and very, we're very supportive of your work. And I thank you for the question. David? You may be muted, David, if you are talking.
Yes, I was muted. Thanks, Jeff. I think that there have been a lot of interest in gene therapy trials for HIV, and a lot of attempts to do this in many different directions. I think if we look back on what was done in the earlier days, we would have real concerns about trying to do a study like that in the current time and current climate, especially the current climate with the FDA. But our trial really examined carefully what everybody else had been done. And what Jeff was explaining to you really is the whole process of making a very good understanding of what had gone before and the little hints of activity and effectiveness that these trials had given. And there are lots of places where individuals who received cell and gene therapy for HIV self-reported the anecdotal information that they felt much better after the delivery of the therapy. They could reduce their doctor visits, many things like that. Those are not solid scientific data in a trial, but when you hear them repeatedly from the participants in the trial, you start to think things were moving in a good direction, but nobody had really solved that last component of it. And the last component really was to do this in a way where we could actually focus on increasing the immune response to HIV. And just taking someone's immune system out and modifying it and putting it back in did not change the balance of power between the person infected and the virus infecting them. And so we wanted to change that balance of power really dramatically. And that's one of the reasons why we believe this product is a major conceptual step forward compared to everything else that had come before. And it really recognizes the power of the immune response to take over and the potential of the immune response to attack any pathogen, any infection, as long as we eliminate the ability of that infection to fight back. And the way HIV fights back is by killing the CD4 cells that are trying to contain it. So we've eliminated that step. And we hope that that is a critical advance and makes a critical difference in the potency and power of these kind of therapies. Jeff, I might also make a response to Angela Pitts here, who has been asking a question, if that's okay with you. So Angela's asking, I believe she's asking about somebody who has a drug-resistant HIV, but maintains solid CD4 count and doesn't have any AIDS-defining symptoms. And Angela, that person, if they have a viral load, a measurable viral load, that person would not be eligible to participate in this phase 1 study. The phase 1 study is very tightly constrained and very tightly defined to give us a look primarily at the safety of the product. And, but we have done experiments with blood samples from people representing a whole range of clinical status, including the characteristic that you're describing, including people off therapy with very high viremia and low CD4 count. And over again, our process has been successful in creating what we think could be useful products from donors that provided blood with all these different characteristics. So while we have to have very narrow definitions for the first 6 to 12 people that are going to go through this study, we have the strong feeling already that it's worthwhile to expand this later and look at a much broader group of people. And designing a product that was useful and perhaps valuable to the largest group of HIV positive individuals possible was part of our initial consideration. We did not want something focused on a very narrow subset of all people with HIV. We wanted the broadest coverage possible. And we just have to slowly and steadily work our way through this problem, convincing the FDA and ourselves at every step that we operated safely and that we see value in this, that we see benefit to address the risk-benefit equation. And then we can begin to open the door wider to more people with varying experiences with HIV.
So David, let me feed you another quick question. So this is from Miami, with Project A&M, would like to know if the trials are open to South Florida patients or only in Baltimore and DC.
Right. The initial approval from the FDA has been to enroll 6 people in this phase 1 study. And we have told them already that we hope that we can go well beyond that in our phase 1. But in order to maintain this control over participants and control over every step in the process, including how physicians speak to their patients about this, how they go through the informed consent process, all of these things are in a very early stage. So we are primarily recruiting only through the named trial sites that we have so far. What you're talking about is a multi-center trial, which would occur for us at a phase 2 or phase 3 level, probably phase 2. And we just have to get through this phase 1 effort first and make sure we're ready to go on to the next stage.
Great. There's a question here from Bala Ernest. Will the patient criteria be the same for commercial use if successful, or is this just mainly for the trial? And how costly would it be in countries like here in South Africa? So David, will you take the first half of that question? Let me take the second half of that question, because that's kind of in my area, the pricing and what we expect to happen with that. But the criteria, please.
Yeah, I believe I addressed some of this just now by saying that you start a program like this with very narrowly defined participants fitting into a narrow range. And then you hope that you're successful in that area, and then you go on and continue to expand. So we don't know right now exactly what the criteria would be for commercial use, but we think it's very probable that it can involve the large majority of people with HIV.
Yeah. I think what's likely to happen is that the constraints will loosen up, and we will also be able to further characterize it during these phase trials, where we'll understand the scope of the patients that are likely to be cured by our process. Now, we have had some early preclinical data that indicated that it was possible to make a suitable cell product from a patient that had a T cell count as low as 200, and that was blood that we should never have received from a vendor we've now gotten rid of that vendor, just to let you know that we don't think patients with that kind of CD4 count should be giving blood necessarily, and we hadn't intended to collect it. But we did get blood with that level of CD4 count, and miraculously it was a little bit more work, but we were able to make a valid blood product out of that. So I think there's a likelihood that the parameters will expand and be more inclusive over time. And as David has stated, as we narrowed it down to sort of the sweet spot of the process and a range that we thought we would get high levels of efficacy. And that's important also to make sure that we energize this project with a good result in the phase 1. But I think you can look forward to it expanding in the future, and that we can find a route for any HIV-infected individual who has not damaged their immune system beyond repair by neglecting their HIV perhaps too long, getting wasting disease or damaging their lymph nodes, that a lot of those should be able to go on antiretroviral therapy for some limited period of time in order to restore sufficient numbers of HIV-specific T cells that they can be rescued, that they can be put through this process. Again, no guarantees that this is going to work, but if it works, I think that we will be able to expand into a broader set of people. Now let me just sort of transition into the economic reality of this. So this is a new cutting edge technology, and as are all cutting edge technologies, they're expensive. And, the price in South Africa on day one is probably going to be the same as the price in the United States. And I don't know what that price is, but I wouldn't be surprised to see it hover somewhere around $500,000 per treatment. But that is not anything that has been officially established. It is just purely speculative on my part, just sort of looking out there at the pricing of other drugs in this category. But here's the thing about technology, and I think it's really important that everybody appreciate this, and it's something that I'm highly sensitized to coming from the computer industry and a whole bunch of technology revolutions that came from that. Microcomputers, software, internet, apps, IT, communications, all those things. Here's how it goes. You've heard of Moore's law, that the processing power of computer chips doubled every year and halved in cost. Essentially a 4x improvement in the power. And what does that mean? Well, over time, things get better and they get cheaper. And the other thing I'll tell you about the gene and cell therapy industry, from my perspective, okay, this hasn't been established by the industry, just credit this to me and blame me for it or credit me with it, whatever. But this is a highly competitive industry. This isn't going to be like the old pharmaceutical industry. It's going to be cheaper to develop drugs, and they're going to be more effective. The value proposition is going to be much higher. The amount of extra life or improved life that you're going to get per dollar is going to just keep going up and up and up. That's that value part. It's going to be competitive because when you're talking about doing tweaks to DNA, it's like modifying the operating system on your computer or creating new software for your computer. There are multiple ways to do the same thing. We don't count on ruling the HIV market. Even if we get 100% efficacy in the phase 1 and the phase 2, we won't rest on our laurels. Because the bottom line is that if we don't obsolete ourselves with a better product at a cheaper price, somebody else will gladly do it for us. I think this industry is going to remain competitive, and it's going to continue to drive down the cost of the value of pharmaceuticals over time. It won't just be HIV, it will be every drug in this industry. And what will that mean? Well, let's think about some other technologies. Very few people in Africa, in Central Africa at least, had landlines, telephones, and none of them had computers. Now, almost everybody has one of these, a cell phone, more computing power than the first IBM mainframes, in your pocket, talks to you, you talk to it, communicates anywhere in the world. That is what happens with technology. You pack more and more value in at a price where suddenly the value to price ratio brings it within reach of even secondary markets and tertiary markets. Okay, South Africa, it may be price restrictive in a way based on the average, annual income down there and the availability of insurance or whatever. But I think the future is bright to get this out to more and more people every year. And I'll tell you right now, we're already thinking about how to make this cheaper and more effective, how to raise the value of it and reduce the cost of delivering it. And that will be reflected in the pricing over time. So we'll get out to those other places. I don't think it's going to happen as fast in uninsured markets and in secondary and tertiary markets. The third world isn't going to be able to necessarily afford it on day one, but I think that the future will allow all that. And my hope is that we'll see the end of HIV globally in my lifetime, or at least it'll be so well managed that it won't be the same sort of fear that it struck into all of us growing up in the 80s. I was in college in the late 70s and the early 80s. And, the only thing as scary as HIV back then was radiation. And so it would be wonderful to put this in the rearview mirror. I think science and industry has done that to a large extent. It's not a death sentence, but this is really the way to handle this. And I think that gene and cell therapy will also offer a competitive pharmaceutical market with a natural technology curve that will drive this thing further and further into the market faster and faster. All right. Let's try another question here. Let's see. After patients give their blood sample of 400 milliliters during the 11 days of waiting, will there be certain things patients should do or not do during the wait time? What do you anticipate the patient experience will be after reinfusion? David, I think that's a question for you.
Yes. So let me give you a little bit more detail about how this works. So the blood sample is what's called an apheresis, which means that we remove the white cells from a portion of your blood and we return the red cells and your blood plasma back to you. So it's a kind of minimal type of treatment just to harvest the cells that we need for making this product. Then it goes into the 11-day manufacturing process that Jeff described. And at the end of that process, we actually freeze the cells in a living state. We can freeze them and store them very safely. They're very safe and stable while we conduct a series of tests on them to make sure they meet all of the safety requirements for being allowed to be infused back into the person. And that could take several weeks after the product is manufactured to complete this rather lengthy list of tests. During that time, the main thing that anybody participating in our trial should do is maintain their antiretroviral regimen, stay on the therapy. If they have concerns about those therapies, they would be in close contact with their regular physician, the person they receive HIV care from. And then when the testing is completed for the product, we ask people to come back in. They'll get a treatment with an old cancer drug called cyclophosphamide, which just reduces the number of lymphocytes in your blood circulation just a little bit to make room for the introduction of the new cells from our product. You get the cyclophosphamide. There may be some side effects from cyclophosphamide. We hope they'll be manageable. And then a few days after that, then we infuse the cells. And then people involved in the study are asked to come back at regular intervals so we can monitor their responses to those cells. We can monitor any side effects that occurred as a result of getting the cell infusion and begin to collect the data that that address the questions we have regarding how stable the cells are and how they're actually behaving once they go back into your body. We're very hopeful about how they're going to behave, including the fact that some of them will quiet down and become very long-term memory and sit there and watch and wait for any time that HIV reemerges as a very, very long-term, now permanent part of your immune system. And we are very confident about the safety of this because we have not changed the general properties of these cells in any specific way. We've engineered them to become resistant to HIV, but we haven't made them more active. We haven't made them different from other T cells. In fact, all of our testing, we can't really find any differences other than the fact that they're resistant to HIV. And so we're expecting them to go back in, be normal T cells, do the job that all T cells are born to do, which is recognize infectious diseases and attack them. And we expect that to occur, and we expect HIV not to be able to stop that from occurring. And people will then have the benefit of a normal immune response to HIV. And if things went perfectly, it would be like having the flu, where you get the flu, you have it for a week or maybe 10 days, and then your immune system takes over, and then it starts to clear out the flu. That would be our most ideal goal for how this would work in the future. And it also may be that your immune system comes into a perfect balance with the virus, where, as Jeff mentioned before, you really don't have much virus in circulation. But if we really, really tried, we could find traces. That would be a great outcome as well.
Great answer, David. Thank you. As a matter of fact, I'd like to throw you right into another one. Somebody asks whether you could discuss the preclinical data that's been done up to this point, the pros and cons. Let me go ahead and give it at 30,000 feet for 1 second and then turn it over to you to give a little bit more specifics. So I think that the things that have been quite confidence building in this project has been the reliability of it. It's, we made an automated cell protocol. That was not an easy thing to do, but we were able to automate in a Miltenyi Prodigy machine, and we could use other machines as well in the future. This is just the initial one that we developed it for, but that it was able to implement GMP inside the machine, inside the bag, basically, that of blood and the bags of reagents that go on this machine, and the culture vessel that eventually yields the cell product. And to do that reliably, as a matter of fact, we found that it was more reliable than doing it by hand in the lab. And that was terrific. We weren't losing too many cells. We weren't damaging too many cells. So, you know, it was a lot of details to get right, but this now is this automated cell protocol that reliably turns out the product. That's a very important and exciting preclinical result. Then it's the level of these cells that we're getting, and the transduction rates are just really off the charts. You know, to get 1 billion of these cells, well, if you ask an immunologist how many disease-specific CD4 positive T cells do you have after you clear a flu or after you clear a cold, they're probably going to say to you 100 million. So think about that. We're going to create 1 billion of that key disease-fighting cell for HIV, and we're going to put them back into the body. They may not all survive the infusion. The body also has a tendency to, you know, it could become a little bit overcrowded. It may eliminate some of them. You know, there's some things to determine in there, but there's a significant margin above what is called memory level of those cells that should be left over in order to fight the disease. So that number is really encouraging. Then comparing that to the Sangamo study is also very encouraging. And then finally, to go to NIH, one of the top HIV labs on the planet at the National Institutes of Allergy and Infectious Disease, and to see you know, a high level of interest there, and to have them want to reproduce our result in their labs was really exciting. And then they were able to repeat our experiment. They were able to show that this process works, that it yields cells that are highly resistant to HIV infection with tremendous ability to clear viremia and latently infected cells. And those experiments are really important in terms of predicting the efficacy. And that is a preclinical result that's also exciting. To tell you the truth, I can't think of many cons. You know, I'm going to leave that for David. David is the guy who has to dot all the E's and the I's and cross all the T's. And so he may be aware of some things that still nag on him in that. But David, would you, you know, like to add anything to what I said, or, you know, put your own perspective in there on that question of pros and cons and your confidence in the preclinical data?
Yeah, this is not purely a technical answer. This is an answer that partly expresses my frustration from working on this problem since 1985. And by 1985, we knew the most important thing that we know until today about HIV, which is it destroys CD4 T cells. And if you just want to say in one sentence, what is HIV disease? It's the destruction of those cells and all the bad things that happen because of it. And yet throughout the research period, from 85 until now, there are very few laboratories that have focused on the problem of CD4 destruction and how to repair it. So people have tried to address this problem by going every route except this route. And the reason is because this route is hard. And it's very, very difficult to work with these cells. People with HIV have tiny, tiny amounts of HIV-specific CD4 cells, unlike any other chronic infectious disease. If you had a chronic infectious disease, you would have 10 to 100 times higher levels. But in HIV, you don't. The virus is still preventing those cells from sustaining themselves in the body, even with highly effective therapy. So the real breakthrough of AGT was to figure out how to overcome that problem and how to grow these cells, right? And so imagine, Jeff's giving you the number of 1 billion, but that means that we can increase the memory CD4 cells in your body by this infusion from between 10 and 100 times, right? And we know that memory cells do this when you're responding to a disease all the time. They increase their numbers, and then what happens later, they decrease their numbers and go into a sort of long-term memory state where they're just ready and in the background, ready to go. And we think that's what will happen here. And we'll create this permanent memory and permanent immune capacity. So why are we confident? Because we asked the question, what is the absolute major thing that HIV does to the body? It wipes out this subset of cells. Why not try to fix that problem? Why go and try to fix 1,000 other problems? Let's just focus on the problem that's most obvious to us. And that's how we got to this point. And it just took a technological achievement and a major effort by the team to be able to get there. So that's what's really unique about the problem. And all of our preclinical data that support this show us over and over again that what we've done is we've created cells that respond to HIV proteins exactly the way you expect them. They have the exact normal response. But when you ask them to do that response and you put in massive amounts of HIV at the same time, they still do the response because they're not being killed. They're not even being infected by that HIV. So now you have a permanent ability to make immunity and the virus can't reverse it. So we have extensive preclinical data. We have looked at blood from about 30 people in total now, and quite a number of people we went on to make full-scale, full-blown clinical products. They meet all of our expectations. They continue to be rather phenomenally enriched in these HIV-specific cells, and the genetic modification is only in those cells. So all the rest of your cells against any other virus or any other bacteria or anything else, all those cells are pretty much untouched. So you have a normal immune system, but just this very special component added to try to get you over the hump in the battle with HIV.
Yeah, it's really remarkable. It just all makes sense. Even if you aren't a molecular biologist, even if you are not a immunologist or an HIV specialist, you probably understand enough about the immune system that you can understand what David is saying and the whole theory and the mechanism of action behind this product. It really makes sense that we have hit sort of the linchpin in with sufficient power using, tools that have really come to the forefront in drug development over this last decade. That we have a good shot on goal here. And I think that, everybody should keep their fingers crossed and hope for the best. But again, I'll just iterate that that's, a lot of the reason that I feel confident about it. I think the preclinical data does show good evidence. I think the arc of all of the studies in HIV cure shows a trajectory, and you can sort of map from the Sangamo study to us that there's been, a major breakthrough during that period. And, so I think that overall the preclinical data is quite encouraging. So I'll roll into a couple of other questions here. This one's just a comment. I was exposed to HIV 2 months ago and almost lost hope. I plan to study computer science next year. This is inspiring. It has given me hope. Thanks for the hard work and explanation. Well, no question there, of course, but thank you very much. And yes, absolutely, do not give up hope. Science and clinicians, scientists and clinicians and companies and the federal government and researchers around the world have really changed HIV from hopeless, horrible, deadly disease to something that's manageable already. And there is no reason for an HIV-infected individual to give up hope. And it is very important that if somebody finds out that they're HIV positive, we highly encourage them to get on antiretroviral therapies if they can get them where they are, and to suppress the virus. And it's painful, but This is something that will lead to a reduction in the spread of HIV, which is an important thing socially. But also it will assure that not only are patients or individuals who find themselves in this situation likely to have a normal lifespan, but it keeps you prepared for a cure that takes this approach. We cannot fortify your immune system against HIV unless you have an immune system, at least a starting point for us, of HIV specific T cells. That's the idea, that once you're on antiretrovirals for long enough, your full T cell diversity comes back. You know that because people stop getting colds and flus and being unable to fight them off themselves. Because what happens when you suppress HIV? Well, the cells that have been depleted start to come back out of the bone marrow again, and they don't get reinfected because the suppressive therapy prevents the transmission from cell to cell. And so people that are well controlled on HIV don't necessarily have the same immune system that they had before the infection, but it's sufficient to fight off normal attack of pathogens. And then even their HIV T cells, even though they're still not useful, they still don't have the power to really fight HIV because they're easy to infect. Well, they're in the mix, and they never see an HIV viron because that's how good the suppressive therapy is, that those HIV T cells are not only there, but they're uninfected. Now, if you go off of your antiretroviral therapy, the unfortunate thing about that is it takes about 21 days for patients to rebound to full viremia and start destroying their immune system again. In the initial acute infection, my understanding is that about 90 days after the acute infection, the HIV will completely eliminate the natural HIV protection. That's how fast it depletes the HIV specific T cells. And why is that? Well, these T cells were born to hunt down HIV. So what's the first T cell that's going to arrive at the pathogen? It's going to be the HIV T cell. And what's going to be the first one to be infected? HIV T cell. I mean, HIV is scary and amazing at the same time, right? It's evolved to just be this incredibly clever virus. It's not only knocks out the protection in the body, but it uses that protection to establish the beachhead of the infection. And it has the ability to integrate itself into cells so that even when they split, they go ahead and they split if they go to the lymph nodes and they replicate the virus. They trade the virus between T cells. Amazing, scary. But okay, that's the key point. And that's what we're attacking at AGT. And so, yes, I'm so glad that being at this press conference may have given some HIV infected individuals hope. This is what you can count on. I mean, we cannot promise you that this is going to work, but what we can tell you is we are absolutely committed, and we are true believers in this approach, and we're going to keep going on this. And I am, like I said, very confident that we'll start to see some efficacy signal in this phase 1. But there are lots of things within this product that can be adjusted all throughout the cell process. There are things that can be dialed up or ratcheted down to change the effect in the body in order to tune this so that it works. There are even things that we can do in the vector. The vector is a series of ACTGs that are coding basically genes or knockdown to genes. And we can adjust those as necessary. So we are going to keep going on this. We think we may have, this project may give us an amazing result that will give you even more hope that a solution is nearby or on the horizon. But we are absolute believers in this approach. We just feel like we have been watching this industry. Our scientists have been in this for decades. And they have seen all of the discoveries, the innovations, the understanding of HIV, and they have really leveraged that in this project. And so hang in there, prioritize getting on ART if you are actually infected. And hopefully we'll get this to you soon. All right, let's look at another one here. What do you predict is the time frame for the first trial to end? 1 to 2 years? Since it's only 6 to 12 people, will phase 2 or 3 be open to in other cities? Okay, so there's a bunch of questions in there, David. What do you think the rollout is of the phase 1? How long is it going to take us to get through the number of patients that we enroll? How many will we ultimately enroll? When do you think this might roll into a phase 2? And what would it take to get into a phase 2?
Yeah, so the study starts quite slowly because of an FDA mandated timing of between individual participants. So it'll start slowly, and then hopefully it'll accelerate as we get closer to the number 6. And then we'll have an accelerated plan for going between 6 and either a total of 12 or a total of 18 in the phase 1 study. If we can make the acceleration, we can complete the dosing of all patients and get safety data from almost everybody within 2021. And so that well before we reach that point, we would have been planning the phase 2 expansion and how we start to collect real efficacy data on how this is working. And so I think that that study wraps up in 2021, and phase 2 would start pretty quickly right after that because of what we would know and how we would be able to proceed. Jeff, I wonder if I could pick up a question that Angela Pitts asked, if you don't mind, another one that she asked earlier. I
Sure.
didn't want it to get...
Yeah, go ahead. Feel free. I have another one after that, and why don't you go ahead on the one that you picked up on?
So Angela Pitts asked us about the range of populations that will be included, and basically it's a question about diversity of recruitment into this study.
That was the same question I was looking at, David. Thanks for grabbing that one. I also have something to say on that subject, but please go.
I mean, in 2000, I gave up a great job somewhere else and moved to Baltimore, Maryland, because if you study HIV, you should study it where it's actually the worst. And so in underserved populations, medically underserved populations, and minority populations clearly are most affected by this problem. And we have located our phase 1 trial sites in exactly these same places. So by working in Washington, D.C., which has the highest infection rate HIV infection rate in the nation by far, and working in Baltimore, which isn't much behind, and these are high proportion minority populations, we think that we're addressing the populations that are most affected. And in fact, we wouldn't do this trial another way. We think that all trials should be representative of the people in the United States. And here we should address the infection rates and how they differentially affect different subpopulations. And that should be represented from the very first start of these studies.
Yeah, absolutely. This is something we're very sensitive to. We're a diverse group of people at AGT working on this. And one of the things that's been brought to our attention is that, and David knew this long before he came to the company, but although Blacks are less than 14% of the US population, they are 42% of the HIV infected individuals in the United States. So think about that. They're bearing almost a 3x burden above other populations in the US. And I think that, this is subject to change over time, but there is an imperative to get out there and to get something that we can get out to all different parts of the US population, and that we should be emphasizing the groups hardest hit by this in order to do the most good in society. And so, David, thank you for your response on that. And yes, we are, going to obviously be studying and pursuing that subpopulation quite intentionally and actively. So let's see here. Do we have any other questions? What is the ballpark timing for patients to return for infusion after the 11-day cell process, both in the clinical trials and at market? I'll just say quickly that we're very pleased that the FDA is allowing us to try this in patients, because remember, well-controlled HIV individuals, HIV-infected individuals who are well-controlled on antiretroviral therapy are technically considered healthy by the FDA. So they had a very high bar of safety that they insisted that we exceed in order to do this trial. And we're really pleased that they saw our therapy as being safe enough to put into these patients. But what they did in the interest of safety is they specified an extensive set of release criteria, testing on that cell product before we put it back into the patients. And that is going to, I've heard an estimate of 73 days to do that release criteria. So even though the cells will be done after 11 days, they will sit in the freezer while we do all that testing and make sure that there's nothing that we can detect in them that could be a problem. And then we will be able to do the reinfusion. We do not expect that that level of release criteria will be necessary in the commercial product. And I think this is a good place for me to turn it over to you, David. What do you anticipate as we move through other phases of the clinical trial and out to commercialization? What do you think the timing will end up being in terms of reinfusion?
I think we already have signals that as long as everything goes well, and these very first few people that come through, we don't see anything unusual, either in their response to the infusion, or we don't see anything unusual in the testing. I think that the FDA is very responsive to data in this way, and we would return back to them, and they will start, assuming things are acceptable to them, they will start removing some of these barriers that are delaying the infusion. And I think realistically, we can get down to a time of a few weeks to 1 month between the end of product manufacturing and infusion. But there are just certain things in there that we can never shorten, and we don't want to, which is checking the absolute sterility of the product and a few other things that are really important for guaranteeing the safety of people who so generously volunteer to participate.
Let me ask you another question, David. I hear a rumor that you have some ideas about how we might even be able to shorten the amount of time of that 11 days, that we might be able to make a blood product more quickly, more efficiently, and even more cheaply than we can do it today. Any, just sort of, doesn't have to be really detailed, but, any general information you can share on that?
Well, I would say that cell and gene therapy of this type, and it's kind of led by CAR T, and Jeff, you'll notice we have a CAR T related question on there too we should address. But cell and gene therapy of this type is in its very infancy. And so everybody is starting basically the way we're starting, with a process that includes the stimulation of cells and the transduction with the vector and the growth of those cells to the right dose. And we're watching very carefully what everybody else in this field is doing. And some people are experimenting with shortening all of these periods, these intervals. And it's quite possible that the time to manufacture can be halved, maybe a little bit better than that. But that's running, and we're still at the stage of walking. So we're looking forward to the opportunity to explore those questions once the trial starts.
Yeah. Any shortening of the process means that more patients can be treated on the same equipment, which means the cost of the equipment comes down per patient as well. So these are all parts of driving down the cost over time. So the timing of these things are important. They're things that we are trying to be diligent about eliminating as much time as we can from all these products while keeping them safe and effective. We have a question here about CAR T. This is the one you were mentioning. CAR T patients are at risk of the kind of cytokine storm we see in COVID-19. Is this an expected adverse event from this therapy? And that's definitely a question for David, but I'm going to say something really layman here just to start David off. And that is that a lot of the cytokine storms that come in CAR T have to do with the unknown levels of tumor burden and the reactivity of the CAR T cells against them, which isn't quite as predictable as the reactions in normal viruses and normal T cells. And that's leading to these cytokine storms where these cells may spin up in the presence of a lot of stimulation too fast and cause a cytokine release syndrome. David, you once told me that, because I had this question a long time ago, you once told me that if we were to reinfuse these cells and they were to attack every single latently infected cell in the body simultaneously and eliminate it immediately, there still wouldn't be enough of a reaction and enough cell debris and enough cytokines where it would be likely to be in the class of a cytokine release syndrome that we're seeing from CAR T. Am I characterizing that reasonably? And could you go ahead and put some more details around this question?
Yeah, you're characterizing it reasonably. And cytokine storm in CAR T we think, has a lot to do with the expansion of CD8 T cells that are specific for attacking. They've been programmed now to attack the tumor with the CAR T molecule. And we have excluded CD8s from our product. So there are still CD8 cells in the person that's infused, but we haven't expanded them or modified them in our product. In fact, we removed them during manufacturing. So that's a protection against risk. We also did it because it makes the manufacturing more efficient. And so we have an expectation based on, as Jeff said, the burden in the body of infected cells, which is low, and the type of cell we're putting in. We have an expectation that cytokine storm is unlikely. But this is a phase 1 trial entering new territory. Our consent forms, our physician manuals, our protocols include extensive information about what cytokine response syndrome looks like, the so-called cytokine storm. Also all the things that are related to that. And we are providing training to our providers so they understand the management of the cytokine storm. And our site in Baltimore is closely linked to the Greenebaum Cancer Center in Baltimore, which has a CAR T specialized clinic where they have very high level of expertise in handling this problem. So we certainly hope we don't see it, but we are prepared to handle it to the best of medical ability.
Yeah. That's a reasonable question, and it really does point out that even if the chances of a problem are small, the FDA and ethics require that we anticipate them and that we have some plan of action if something like that crops up. So, that's the situation. We think the likelihood is very low, but we, certainly are thinking about that issue and anticipating it in the study, even though we don't believe it will turn out to be one of the adverse events that we see in the phase 1. This is an interesting one from a medical student. What about latently infected cells, especially in the brain? David, I mean, you really know the pathology, and the virology of HIV. So how far does it spread in the body, and where can these persist, and where can it be eliminated by this immunoregulatory approach? And what would be the effect if there still was some latently infected cells outside of the reach of these T cells, if there is such a place?
Yeah. The infection in the brain has been a concern for a very long time. It's related to a condition called AIDS dementia, which thankfully is very responsive to antiretroviral therapy. So when the therapy is given, that problem subsides to a large degree, but it doesn't go away. And there's a lot of controversy about how those infected cells are maintained in the brain. So we are attacking the target that's accessible to us, which is to put memory CD4 into the peripheral blood and let it distribute in what's called the secondary lymphoid system. It's possible that some of those will go to the brain. It's possible that activated CD8 cells will go to the brain and kill the infected cells there. But we believe that the brain reservoir of virus largely requires regular replenishment from other parts of the body, other compartments. So if we can stop the virus replication everywhere else, we should be able to allow the brain infection to subside. We hope that that occurs. That's the prediction from the majority of literature, and we'll be watching that down the road as we get into more detailed clinical studies.
Thanks, David. This one's an interesting one also. Could you say more regarding the potential for autoimmunity, another effect of CAR T, which we're keenly aware of? It's one of the reasons you may not be aware of this listening to this press conference, but we also have an immuno-oncology asset. Hopefully we'll be back in a press conference about that one day when we can start a clinical trial there. But we're very aware of some of these issues in CAR T. And again, the fact that we're working on T cells, we've kept that in focus. So this person's asking, is that an autoimmunity possible? Let me again give the CEO response and then turn it over to David. One thing that's important to remember about the modification that we're making to these T cells is that it does not change their behavior whatsoever. So we know what the effect of not having CCR5 has on T cells, because there's many people in Northern Europe who are born with that depletion. They're born without those CCR5 receptors, and we know what the side effects of those are. And so, the side effects happen to be that the CCR5 receptor is useful in crawling through tissue, and what the patients experience, or the people who have that genetic mutation experience, is that skin bacterias and funguses can be a bigger problem for them. And I think a lot of people would certainly be willing to go ahead and trade HIV immunity for having to put antibiotics and fungal creams on their skin, if their CCR5 was missing. So I don't think those patients are or those persons are necessarily in a really bad state. But that's the effect of the modification we're making to the exterior of the cell. The siRNAs that we're putting in there have been run against all known messenger RNAs, and it doesn't look like there's any chance that there would be a match against anything but HIV. So those siRNAs are unlikely to affect anything that is non-HIV genome. We're not putting another antigen receptor on these T cells. They are born with the right antigen receptor already, and they only need one, the one that is sensitive to the HIV pathogen. So we think autoimmunity is significantly less likely to occur in this than in a CAR T product that has bispecific T cells, because they're that's sort of an obvious potential consequence that the new CAR might target something unusual, or that these might cross-react and target something. But our feeling is that there's not a high potential for misdirected T cells that might cause a new autoimmunity in the patient or in the individual. David?
No, Jeff, you've said it just right. Doesn't need anything from me.
Thank you. I feel quite proud of that, David. Thank you. All right, well, let's see here. Do we have any other questions? So I haven't seen anything come up in the feed that were the selected questions. There's a couple editors in the background on our side that are picking out questions, but if you have any that I missed, could you just throw them up there again, and I'll go ahead and take them. I think, you know, we could go another 20 minutes in this press conference and still be running the time that I had scheduled on my calendar, at least. I don't know what we announced to folks out there in the audience, but I think we'll go another 20 minutes if there's any more questions. Got anything for me, Norman or Sasha? Well, here's another question really along the same lines, and I don't really understand lupus myself, but the question is about cell attack. Could this somehow create a lupus attack? David, do you have any understanding of that?
Well, that's sort of what you were just talking about before.
Mm-hmm.
So we can't, I mean, I can't, let's not, you know, I can't imagine how this would create a lupus-like attack.
Mm-hmm.
Create something that feels a little bit like that for a brief time, because that lupus attack and what we were speaking about before, cytokine release syndrome, or the problems from CAR T, they have some overlap. But again, it would be, if it occurs, it should be very transient, because there's a big difference between programming a T cell to have a new way of being stimulated, which is what you do for CAR T cancer therapy, and programming a cell just to resist HIV. There's a world of difference between those two things. And we don't program the cell to have a new way of being stimulated. So we expect it to behave normally. If you didn't have lupus prior to us doing this process, we don't expect you to gain it through this process.
Okay, we have a couple of other questions here, but it looks like we may be wrapping up soon if we don't get more questions from the audience. One was, how will this treatment be administered to patients? I think the main question is, you know, how does it get into the body after it's created? That's by infusion. So, you know, you've seen these done in hospitals. They're common with chemotherapies. It's the same as a CAR T infusion. There will be a mild cytoablative, not myeloablative, but cytotoxic treatment that's given beforehand. There's a theory that you need to clear out a little bit of room, because during that 11 days, or certainly during 90 days between the blood draw and the reinfusion, the body will have recovered sort of a normal number of T cells in the bloodstream, and the body doesn't like to have an overcrowding. So if you do crowd them, the body will eliminate them. So it is normal when you're reinfusing T cells that you go ahead and you put in a chemotherapeutic that will eliminate some of the T cells in the body to make a little bit more room, so the body won't feel overcrowded when you do the reinfusion. And then it's just a normal infusion. So that is the way the treatment is done. 400 milliliter leukopak, 11 days in an automated cell process. And then it is, as David said, cryopreserved while we do extensive safety testing and release criteria. And once we've determined that that dose passes all of those release criteria, the blood is defrosted. It's restored to normal temperature. The cells are preserved very nicely that way. And then it is just a reinfusion, basically. That's how it's delivered. And let's see here. Will the trial participants be required to go to a clinic or a hospital to receive their therapy? It is all done on an outpatient basis. And in fact, even during this study, this initial study of it, it's not required that the patient stay in the hospital. You may be familiar with CAR Ts that it is a requirement that there be a hospital stay. In our case, they can come in for a leukapheresis and go home. and then they can come in for the cytotoxic treatment. And I think there's a little bit of time between that and the reinfusion, so they won't stay over in the hospital then either. And then they'll come in and get the reinfusion. None of those things will require a hospital stay, but they will all be done in a clinical setting. Especially in the United States, in this study, they're going to be done under clinical conditions that are appropriate for the type of antiseptic environment and the kind of services that need to be delivered to the patient during this therapy. So, David, anything to add on that?
No, that's good.
Okay, I got to pass on that one too. David was a professor, by the way, of all these things. So some of his grad students may be on this call for all you know. All right. Can I assume that trial participants must be HIV positive to be eligible for the trial? Yes, you absolutely can assume that. Let me tell you one thing about gene and cell therapy. It is still cutting edge. It is incredibly powerful, and it has it has a significant tail end, right? When you use a lentivirus to make a modification in something in the body, and when you're modifying T cells, they can persist for decades in the body. This has already been demonstrated. So it's not something you can undo easily. And that is why the FDA has restricted gene and cell therapy to acute needs. And so it's one of the things that actually is a benefit in terms of speeding these things through the clinic. It makes a slightly higher bar to get into the clinic because the FDA has to be satisfied from preclinical data about the dosing and the safety and so forth. And all they've seen is animal models and toxicology studies and stuff like that. So there's a lot of, it takes a lot of deep scientific logic to decide when something meets that bar. It requires quite a bit of expertise to supply that data and even to evaluate that data at the FDA. But once they decide that it's ready for the clinic, there is not an option of putting that gene or cell therapy into a healthy person. This is not the kind of thing that goes away after a day or two. It doesn't have a normal pharmacokinetic or pharma distribution profile where you can assume that if somebody gets sick, well, the effect of the drug will go away over time. It could be permanent. So there's no, as far as I know, there's been no gene and cell therapy that has been prescribed by the FDA to be put into healthy patients. Now, the benefit to the company and the benefit to the market is that the phase 1 is done in patients that have the disease. So you almost always get some sign of efficacy because you're treating people that have the disease, and you get an opportunity to see what is the effect on the disease, as well as determining whether the basic dose is in fact safe. So David, do you want to add anything on that?
No, that's fine, Jeff.
Cool. Wow. I'm batting 3 for 3 on those. What does the therapy do besides CCR5 knockout? David, I kind of covered this before, but would you go ahead and give a quick answer on, what are the things that we're doing that give a high level of protection to the HIV specific T cells so that they can get back in the body and do their job of actually clearing and controlling the pathogen?
Right. CCR5 is only one small part of the HIV infection story in the body. And removing CCR5 does not eliminate the ability of HIV to grow, to enter that cell. It just takes a slightly altered form of HIV that uses a different molecule instead of CCR5, but it can still get in. And that type of virus, called a CXCR4 virus, is prominent in late-stage HIV patients, especially in North America, and can be quite devastating to your immune system. So just knocking out CCR5 is not a complete story in any case. And very importantly, knocking out CCR5 does not prevent a cell that's already infected from releasing virus. And so in the reality, in the real situation in the body, people are carrying some already infected cells. Those already infected cells are quiet and the ability to release virus is controlled by their drug therapy. But if the drug therapy was removed, those cells would reactivate and start to release virus again, triggering this kind of wildfire of cell growth and virus production that leads to the destruction of the immune system and resets the clock way back to the earlier times in disease. So you have to be able to control several different aspects of the function of those cells. You want to make sure they can't be infected by the CCR5 viruses, make sure they can't be infected by the CXCR4 viruses, make sure if they already have a virus, it can't come out of them. Now we can't remove CXCR4 on the surface of these cells because it would destroy some of the required functions that we need for them to perform. So we only can remove CCR5. That's the only target we can actually take away. And then we put in these other microRNAs which target parts of the virus itself that can stop the X4 virus or the R5 virus or virus that's already in the cell from being produced and coming out. Okay, so the whole idea is that now that CD4 T cell, when it sees virus, it can be stimulated, proliferate, and do all the functions that it's supposed to do normally. So the second part of that question then is, what are those functions that we expect it to do? So one thing is, we know that CD4 cells can attack virally infected cells on their own. And in many people, this is a very important part of the immune response that's gone when HIV removes those cells. Secondly, CD4 T cells are required for rapid production of new killer T cells. This is the dominant way your body goes after and finds infected cells and destroys them so they no longer provide putting virus into the system. Those killer T cells can be escaped by mutations in the virus, and you need to make new ones all the time to keep up with the mutation, with the viral mutation. If you don't have a strong HIV-specific CD4 response, you don't do that function. That mechanism doesn't work. And so the virus just gets ahead of the immune system by mutation, and the immune system can't keep up and fails. And the last thing is that strong CD4 T cell responses are required to support the very best kind of antibodies that you can make. And persons with HIV very often make a lot of antibody against the virus, but they don't make an antibody that blocks infection. And we think if we can increase the amount of CD4 T cells, providing what's called help in the immune system, that we will even promote the ability to make potent neutralizing antibodies. So other people have raised some issues about vaccine. Is this a vaccine? Could this be a vaccine? This has every characteristic of a therapeutic that you would want, being able to fight the virus in real time, and it has every characteristic of vaccine that you would want, being able to prevent you from being susceptible to infection in the future. So, so if we get it right, we get a home run, and we get lots of things accomplished at the same time, all through restoring this critical cell population that the virus took away.
So there's a couple questions here that I think are really interesting and important. Let me start with this one, just because I think that it has to do with sort of further stages of testing. What will happen with the antiretroviral treatments for these patients that are in this study? Will they stay on their antiretrovirals, or will they go off their antiretrovirals? And sort of what is the expectation in future trials, and when might those happen? So the answer is that in the initial study, for added safety, we agreed with the FDA to not require a treatment interruption. So these patients will stay on their antiretroviral therapy. They will not be required to go off of it. In past studies that have been similar to this, sometimes patients have voluntarily, in consultation with their doctors, decided to go off of their antiretroviral therapy. There are studies that say that treatment interruptions are not dangerous in terms of long-term health of the HIV patients if they're short and if they get back on their antiretroviral therapy before the viremia rises to a damaging level. But in our initial study, we will not be requiring a treatment interruption. David, what will come next? How will we get the gold standard to show that these people are able to maintain their viremia without antiretroviral therapy? What's the steps to get there?
Steps, first of all, for us to understand how the durability of the modified cells that we put in. We put them in, and do they stick around as we expect? The second is we should be able to see them creating a new level of T cell memory against the virus at a much higher level than was in that person before we administered the treatment. That's a very, very strong sign that something has been corrected. And we'll continue to collect these data, collect the safety data. And if the procedure is very safe, and if we see clear signs of a change in the immune status of the individual, then we will present this to the FDA, probably in a phase 2 application, and say, we think now we have a reasonable basis for asking some people to discontinue their therapy to really test the effectiveness of the treatment. I think that you want to do that be very cautious about that. We want to be conservative about that. But we also understand that brief interruptions of therapy really don't have a consequence for long-term health of individuals. This has been studied in quite a number of clinical trials so far. But it's a very important decision. It's a kind of difficult decision to take. And we want to be absolutely convinced, and our data safety and monitoring board for the trial, which is an independent oversight board that watches what we do, they will want to be convinced before we even bring this up in a discussion with the FDA.
Okay, so I am going to give 2 more quick questions in the last couple of minutes that we have here. Could this technology lead to a vaccine? This is not specifically oriented towards a vaccine, but we believe that we have some technologies that we may be able to leverage within AGT to help to progress vaccines. David, do you have a quick answer to that? Do you want to express any sort of thoughts on where we might go and how this might lead to a vaccine?
Yes, Jeff. So as we've talked about many times within the company, we have really long-term aspirations for this project, and we don't want to stop until this becomes a practical therapy that can be given on mass scale. And right now we're in the very first stages of it. And why do we have to be at this stage? Because we're in unproven land. Nobody has tried to focus on modifying HIV-specific CD4 T cells. That's not been done as a priority anywhere else. Once we establish that that's really the fundamental mechanism, it leads to a lot of other things. Can you imagine this kind of approach going into a vaccine? Yes, you can. Or you would stimulate somebody's immune response with a normal vaccine, any of the zillion candidates out there, and follow up by modifying those cells so they can't be eliminated if they see HIV. We know exactly how all the vaccines have failed to date. There's very intensive studies on how and why vaccines have failed. We know what it is, and it's because you can't stop that first attack of HIV on this precious cell subset.
Yeah.
And so yes, 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 and yes, better technology to cheaper approaches, to wider distribution, to applying it to vaccine, yes, to all those things.
Terrific. Yeah, no, I think that in part of that answer is that in AGT, you have a company with the expertise and the focus and the will to try to eliminate HIV in our lifetime. And that, of course, involves all sorts of next steps, all the ones that David just said, driving this cost down and getting the cure out to more people, and then finding a way to immunize people in vulnerable communities so that the disease spreads less, that's going to involve a vaccine. And we have an amazing team here in terms of every aspect of the business. And we found a model for developing science, for developing drugs, for developing markets that we think can do this in a cost-effective manner where the public will really be getting good value out of the drugs that we'll be producing in terms of the real world effect, longevity of life, and quality of life that will make these drugs worthwhile, that will reduce the overall cost of healthcare in the world, and that has potential to make a pivotal difference in some of these diseases. And we're at 3 p.m., but I want to tease everybody out there with one more thing. David, HIV in some respects could just be considered a chronic viral infection. Aren't there other chronic viral infections out there? Do we have any technology that might be useful in that area? So could that be a follow-on thing to HIV?
Well, I can continue saying yes.
Oh, the answer to that's yes. I would never have known. We talk about this a lot around the company, and you know, HIV is our first focus, and it's really taking all of our energy, and it's a ton of resources to push this thing forward. And it's been, you know, it's been quite an effort on all levels of the company. And but if we can make a breakthrough in HIV, there are a lot of different technologies that we've developed that have to do with isolating a particular type of T cell and making it better at fighting the disease that it was intended for. So in the same way that we're going to make HIV T cells that are basically just better at their job, one day, I think you can look forward to us building on that same platform, changing out the T cell that we're going to isolate and modifying it differently in order to start to clear other chronic viral infections. We think that HTLV is an obvious one, but this could extend to hepatitis B, human papillomavirus, herpes, and things like that. This is the thing I love about gene therapy. It's one of the things that really keeps us motivated around AGT. We founded this company to make a difference in the lives of our fellow human beings, and there is tremendous opportunity to do that. This press conference is around an event that it was just so exciting for us, and we're so happy that you're sharing this with us, and we really appreciate you coming to this press conference and listening to this information and following AGT on our, sign up for our newsletter, follow us on social media. You know, hopefully we've given you a really accurate picture of what's going on so you can spread accurate information about what we're doing. But, you know, it's a pivotal moment in moving forward on something that we've been working on and dreaming of for a long, long time, to have the FDA say, okay, you can go forward into the clinic. And now we are, you know, we were ready to hit the ground running, and we're going to get that thing kicked off as fast as possible and try to get results as quickly as possible in this phase 1 so that we can hopefully prove that HIV is curable. And if that works, well, the great thing is our work isn't done. It's not just that we'll go ahead and, you know, keep progressing towards commercialization of an HIV cure. It's that we'll reapply all of the things that we've learned about chronic viral infections and gene and cell therapy and all of the tools and components that we have developed around this company that are useful in a wide variety of diseases to keep on knocking out other diseases. And we think that we can do it in a way where the efficiency of developing these drugs is so high that our ability to do it is good, and the cost of doing it is reasonable, that there are many, many diseases that affect millions of people that we may be able to go on and continue to pursue and to continue to make breakthroughs in. So with that, I will go ahead and wrap up the conference. David, do you have any closing remarks that you wanted to make?
No, I appreciated all the great questions and the discussion.
Yeah, let me second that. I appreciate everybody's attendance, all the great questions, all your patience to listen to all this information, and we'll wrap it up here. So we appreciate it if you'll keep good thoughts and your hopes and prayers around this project, and continue to support us in the way that so much of the world, from investors to advocates to scientists to clinical advisors to marketing people to everybody within the extended AGT family. We hope that you'll continue to think good thoughts about this project and others, and we hope we'll see you again soon with additional news. Thank you very much for joining us today. Bye-bye.